We've discussed now how to actually put together a polypeptide. That's by linking together amino acids through peptide bonds. But that polypeptide is not functional inside the cell. It's not going to become functional until that protein is folded up in the correct three-dimensional shape. As we see on this slide, these are just a couple of models that depict how proteins are folded up or what they look like once they're folded up. These would be examples of what we call a globular protein. So this protein forms kind of a glob or pretty spherical structure. But proteins can take on all different types of shapes. So globular proteins are just one example. We do have some proteins that are more stretched out. We have some that are more box-like, some that are more like a barrel or a cylinder that has an interior compartment. So we want to go through how these proteins get to that three-dimensional shape. This picture right here is just showing you some of the importance of a protein having the correct shape. Here we can see that we have a virus protein, so from a flu virus, and then there's an antibody protein which is supposed to match up pretty much exactly with the viral protein. Without a correct fit here, it's like a lock and key fit, without the correct fit, then we're not going to get the desired immune response that we want inside of the cells or inside of the body for that matter. So cells will expend quite a bit of energy and effort in making sure that their proteins do get folded up correctly, because if they do not, then they have completely wasted their time and energy. So there will be 4 levels of protein structure. We're going to call them primary structure, secondary structure, tertiary, and quaternary structure. And the primary structure is really what we've already talked about. So the primary structure is hooking together the amino acids in the correct sequence. So we do have 20 different amino acids that can be used to construct our proteins. And there will be a specific order that those should be hooked together. Now that order comes from our genetic information. So that is essentially what our DNA specifies for our cells. Our DNA tells the cells what order to hook together the amino acids. Keep in mind that in some cases these proteins are going to be several thousand amino acids long, and each amino acid has to be in the correct place. Now, no matter how long this polypeptide gets, as we pointed out before, there will be one end that has an amino group and the other end is going to have a carboxyl group. So when we talk about the primary structure, that is the formation of the peptide bonds through dehydration reactions to give you the long polypeptide. It is not going to be a functional structure at this point. Now as we move on to the secondary structure begins the folding process. With the secondary structure, it will specifically be the backbone of the molecule that is involved. So by backbone, we are excluding the side chains from this. So the R groups or the side chains will not be involved whatsoever in the secondary structure. And with the secondary structure, the molecule will begin to fold up, either by kind of folding back on itself, as we see in this example, or by coiling up, which we see in the bottom. Now, with the secondary structure, it is going to be due to a lot of hydrogen bonding. If we look at this molecule here, we do have a lot of polar covalent bonds when we look at the backbone. If we look here, this would be a polar covalent bond. The oxygen would be a little negative, the carbon a little positive. If we move on, this nitrogen would be a little negative, this hydrogen a little positive. And if we go on the other side and label a little bit, we can see that if we fold back, there will be many examples or areas where we can get some hydrogen bonding take place. So the dotted lines in this slide represent hydrogen bonds that have formed, and it is hydrogen bonds that will hold together the secondary structure. When those hydrogen bonds are formed, we're going to get 2 different types of secondary structure. We have what are called beta pleated sheets, and that's what the first picture or the top picture is representing. So notice that it has folds in it, like pleats would be. And then the bottom picture is representing what we call an alpha helix, and that's going to be a coiled structure. Once the secondary structure is formed, That's where we then go on to use those side chains that are hanging off to the sides. This is a tertiary structure. The tertiary structure is really going to start to put it all together so that we get that three-dimensional shape. We're going to be involving the R groups or the side chains, and keep in mind that there is a lot of variety when we have the side chains. So we have 20 different side chains possible, and this is going to mean that we're going to have a lot of different types of bonds possible here as well. So we could very well have hydrogen bonds involved. We could have some ionic bonds involved. In some cases, we'll get some covalent bonds, such as a disulfide bridge. We can also get what's called hydrophobic interactions. Hydrophobic interactions are just grouping together molecules that are both hydrophobic. And what that does is it gets them away from any hydrophilic groups that might be involved. Now, when these R groups interact with each other, it is very specific how they should interact with each other. So this means that this would specifically be, say, amino acid number 5, maybe its side chain, and maybe this is amino acid number 150. And those two side chains are supposed to hydrogen bond to each other. If they form hydrogen bonds with some other side chain instead, then we may very well not get this three-dimensional shape that is the goal here. So it's pretty amazing that if you have a polypeptide that's made up of maybe 400 amino acids, that they could all fit together in the perfect way every single time. But again, the cell does spend quite a bit of effort in making sure that that takes place, because if it doesn't, then this whole protein is going to be useless. For many proteins, they will only have 3 levels of protein structure. So once they have finished forming their tertiary structure, they will be done at that point. For those proteins, they're then ready to go out and function inside the cell in whatever role they are destined for. For other proteins, they will go on to form a quaternary structure. The quaternary structure is only necessary when you have multiple polypeptides that are going to assemble together in one functional unit. So there are a couple examples of that on this slide. Hemoglobin is a protein that's made up of 4 individual polypeptides. Those 4 individual polypeptides are what we call 4 subunits. They will each go through the primary, secondary, and tertiary structures on their own, and then they will all be put together in this larger complex, which then we call the quaternary structure. For molecules that require a quaternary structure, they will not be functional until they get that quaternary structure achieved. So just to review it all, the primary structure is just making the polypeptide. This is going to involve dehydration reactions that make the peptide bonds, and the information for that is going to come from the DNA. With the secondary structure, it will be hydrogen bonding, and that will be with the backbone. With the tertiary structure, we're going to have the R groups involved, and this will be various bond types. And then with the quaternary structure, it's going to be multiple polypeptides. You want to make sure you know what the difference is between the different levels of protein structure. If we look at this tertiary structure, perhaps you can get a little bit of a better idea of what's going on here. The secondary structure would have formed this alpha helix or this beta pleated sheet or the one right next to it. But what the tertiary structure does is it ensures that, say, this beta pleated sheet is going to be right next to this one, or that this alpha helix is going to be on this end of the molecule. So it kind of orients everything spatially. If we look at an example of what can happen when proteins do not get folded correctly, sickle cell is a very good example. And we're going to look at sickle cell disease numerous times throughout the semester to illustrate some different principles that we'll be talking about. Sickle cell disease is caused by a slight change in primary structure. So in other words, the correct amino acids are not linked together. In this case, it's one minor change. In this picture, we're seeing someone that does not have sickle cell disease. So for this one, here would be the proper primary structure. That's going to fold up with the secondary and the tertiary structure, so we get this shape. And then that actually has a quaternary structure, so we get this larger structure. And then inside are red blood cells. These molecules are going to be floating around in there. They're going to remain separated from each other. For someone that has sickle cell disease, there is one amino acid that is incorrect. Keep in mind that the primary structure is determined from the DNA. So this is a genetic disease. So that number 6 amino acid is slightly different from what it ought to be. So in other words, the side chain is different, which means it won't be interacting with perhaps the other side chains that it's supposed to. When the secondary and tertiary structure is formed, we get a slightly different shape. The quaternary structure still forms, but the problem with this new shape is that we have this one hollowed out area right here, which is going to be hydrophobic. Hydrophobic things like to stick to other hydrophobic things. What this means for the cell is that all of these individual quaternary structures begin to clump up. When they clump up, it alters the overall shape of the red blood cells so that they become sickle shaped. And that's where the name of the disease or disorder comes into play. Normal red blood cells are supposed to have a disc shape to them. With the disc shape, they can move very smoothly through our veins and arteries. If you have the disc shape, then we've got some jagged edges on there which can certainly get caught on things. So that does become an issue, especially when the blood needs to move through the veins and arteries very quickly. So this example is just to show you that sometimes it just takes one minor change at the very beginning of everything in the primary structure, and it alters the rest of the shape of the protein. Although it may look like it's a very slight alteration, it does change the function of the protein or the way it interacts with other molecules inside the cell. Now, because the shape of the protein is so important, that shape is going to be dependent on the physical and chemical environment of the protein. So by chemical and physical environment, we're talking about things like temperature, pH, other ions or molecules that might be present in solution. And every protein is going to have really an ideal environment. If that ideal environment is altered, then that could change the shape of the protein. So denaturation is the process where a protein that is perfectly folded up does happen to get unfolded. So that is not a good situation for a protein. So here on this side of this diagram, we have a normal protein, or what might be called a native protein. This one is folded up and it's functional inside the cell. If the conditions changed, so this might be something like an increase in temperature or a change in pH. This causes that protein to be denatured or to unfold. So we're disrupting hydrogen bonds that may be holding it together. We may be altering ionic bonds, hydrophobic interactions, but in any case, our protein is not held together, and now we just are back to the primary structure, that floppy polypeptide. That is not functional inside the cell. So what we've done is we've taken a functional protein and we've destroyed its function. In some cases, if the conditions are returned back to normal, then that denatured protein is going to be able to refold. In some cases. In other situations, that denatured protein will not be capable of refolding. So if it cannot refold, then that protein is essentially garbage. The cell will have to completely take it apart, discard it, and then rebuild a functional protein in its place. Now, some proteins are able to fold on their own, and they will go through several steps, perhaps, as they get to that very perfect, folded, stable conformation. There are other proteins, however, that need some help. So we have proteins inside the cell whose very job is to help or assist proteins with their folding. These are called chaperonins. So if you think of the term chaperone, these are going to guide the folding of other proteins. There's a whole group of them, so we're seeing one type kind of shown on this slide, And this particular chaperonin has a barrel structure to it and a little cap. So there is a compartment inside, and a polypeptide that is unable to form its proper structure is going to enter inside the chaperonin, the lid will close, and then the protein is going to fold up. At that point, the lid comes off, the folded protein goes out, it can go and do its job wherever it needs to in the cell, and now another protein or another polypeptide can actually enter and fold up. So in this case, at first it may seem like there's nothing special about the inside of this chaperonin that now suddenly that little protein can fold up inside. However, what the chaperonin is able to provide is the perfect environment. Now that may be different for different proteins. But notice that out here in the cytoplasm, there may be other proteins that are trying to fold. Those proteins are going to have side chains, which may be interacting with hydrogen bonding, ionic bonding with this one right here. So if we have all these other interactions, we may be affecting its ability to fold. So perhaps it just needs isolation. So by entering the chaperonin, we're isolating it so that other factors are not going to be affecting it. It may need a hydrophobic environment to fold up. In that case, we're not going to find that just floating around in the cytoplasm of the cell. So we put it in here, we provide the hydrophobic environment. Whatever that protein needs, it's finding in that environment, and once it enters, it can fold up and it gets to a very stable conformation, which it can then hold when it exits. Chaperonins work on a subset of proteins. It's not a one-size-fits-all by any means, but they do work on more than just one specific protein. There are a lot of different chaperonins inside the cell. If you have a protein that requires a chaperonin for folding, it would have a very tough time refolding itself if it had been denatured. So those are a lot of the proteins that we say are not capable of renaturation or becoming functional again if they were to get unfolded. Now you might be wondering how we actually know that the chaperonin has a barrel shape to it, or how we would know that a protein has a globular shape to it. There are specific techniques that are used to come up with protein shapes. So I'm just going to introduce these two techniques to you. We're certainly not going to go through them in detail. These are really techniques for advanced biochemistry classes and things of that nature. But just to give you an idea, one way that protein shape is determined is through X-ray crystallography. And with X-ray crystallography, essentially a crystal is made of the protein. And what that means is that you have a solution that contains the protein. That solution is dehydrated, so we get rid of all of the water, so that those proteins are now in a repeating arrangement, which is the crystal. An X-ray source is then shown on that crystal, and the beams are deflected in some manner depending on the angles of the protein. So there is a detector. There are a lot of computer programs that are used to help interpret the shape or the pattern that is made from the X-rays, and then that's used to come up with an overall shape of the proteins. Very complicated technique. Another technique is what they call nuclear magnetic resonance, or NMR spectroscopy. And in this case, it does involve the use of a very high-powered, very large magnet, and that magnet affects the spin of the electrons that are involved in the formation of the protein structure. And again, with a lot of computers and so on, that information is used to come up with a three-dimensional structure. So do know that a lot of protein structures have been determined using various techniques. So it's not all complete guesses as to what these proteins look like inside the cell. The last group of polymers that we're going to talk about in Chapter 5 are the nucleic acids. And while they're the last group of polymers we're going to discuss, they are also by far the largest of all the polymers that you find inside the cell. So these are going to be macromolecules composed of smaller subunits or monomers, which we call nucleotides. Because the monomers are called nucleotides, the polymer is going to be called a polynucleotide. We say that they're very, very large because in this case, we're talking about linking together as many as 200 million nucleotides. These are going to be responsible ultimately for determining the primary structure of a cell's proteins. So these will form the blueprint or hold the information that the cell will need to actually make its working molecules. We have two examples of nucleic acids. They are deoxyribonucleic acid, which is known as DNA, and then ribonucleic acid, which is known as RNA. So we want to talk about the nucleotides. The nucleotides, which are the monomers here, they're going to be composed of three parts. They have a nitrogenous base, and we have a few different nitrogenous bases that can be in that position. They are made up of a pentose, and a pentose is a 5-carbon sugar, and then they're going to have a phosphate group as well. So if we talk about these different components, for the nitrogenous bases, there are 5 different possible nitrogenous bases that can be present in a nucleotide. These nitrogenous bases fall into 2 categories. We have what are called the purines and the pyrimidines. The purines are the larger ones, and these are basically double ring structures. And there's 2 possibilities. We have what's called adenine and guanine. We usually refer to them with just the letter that starts their names off. So this is A and G. And they do have different structures to them. You're not required to memorize those structures or recognize those particular structures. But we should know that the purines are double ring structures and the adenine and guanine are examples of them. As far as the pyrimidines are concerned, the pyrimidines are going to be smaller, and these are going to be single ring structures. We have 3 examples of pyrimidines. We have thymine or T, cytosine, which would be C, and then we also have uracil. Now, depending on whether the nucleotide is to be found in DNA or RNA, the nitrogenous base that you find is going to be a little bit different. So in DNA, we can have A, T, C, or G present. And if we're talking about a base in RNA, that's going to be A, G, C, or U. So what it boils down to is the T and the U. Thymine will only be found in DNA, and uracil will only be found in RNA. We look at the sugars. All nucleotides will have a pentose sugar. The 2 possibilities for the sugar are very, very similar to each other. In RNA, we have what's called ribose, and in DNA, we have deoxyribose. And the only difference between the 2 is the presence of this hydroxyl group in the ribose and the absence of it over here in the deoxyribose. The sugar is where the names DNA and RNA come from. So DNA, the D stands for deoxyribose, and the R in RNA stands for ribose. And that one difference is enough of a difference that it is going to alter the overall shape of the molecule. So DNA and RNA will have different functions inside the cell. Also want to point out that when we talk about nucleic acid structure, there's quite a lot of talk about the 5' end and the 3' end. Well, what this 3 and 5 are referring to is they're referring to the numbers of the carbons in the sugar. So this would be 1, 2, 3, 4, and then we have 5 up here. Notice that the 5' end is attached to the phosphate group, and the 3' end is always going to have a hydroxyl group on it. So whether we're talking about ribose or deoxyribose, both of them do have a hydroxyl group on the sugar. So we will have 2 distinctly different ends to our nucleic acids, and we call it the 5' end, which is the phosphate end, and the 3' end, which is the hydroxyl end. The amino acids will be, the nucleic acids will be constructed by hooking together the nucleotides. These nucleotides will be joined together with linkages called phosphodiester linkages. And when the phosphodiester linkage is formed, we're basically taking the 3' carbon of one and we're hooking it to the 5' carbon of the next. So this 3' carbon right here, that would get hooked to a 5' end of the next. So if we just label these phosphodiester bonds, here would be one, here would be one, here would be one, and so on, down the molecule. Now we may be hooking together millions of these. The difference between the nucleotides would really be the base that's present. So whether it's an A, T, C, G, or we talked about U's being present in the RNA as well. So that's the unique part to the molecule. Notice no matter how long we get, we're always going to have one end that has a 5' hanging off of it and one end that has the 3' sugar hanging off of it. Three-dimensionally, we're going to have DNA molecules that are a double helix. So you've probably heard of a double helix or seen a double helix of DNA at some point in your life. And here's just a quick look at it. The double helix was discovered by James Watson and Francis Crick in 1953. They did win a Nobel Prize for that work. And what the double helix is saying is that you've got two strands or two polynucleotides of DNA, and they're going to actually be hooked together. They will be hooked together like a ladder, and then they will be twisted. It will be hydrogen bonds between the bases that actually hold the two strands together, and that means that the sugars and the phosphates will be alternating down the outside. So notice we've got sugar, phosphate, sugar, phosphate, sugar, phosphate, and so on the whole way down. Additionally, the chains are going to run in opposite directions, or they will be what we call anti-parallel. That means that the 3' end of one is going to be across from the 5' end of the next. And we have the same thing on the other side of the molecule. Be familiar with the general structure of DNA, the fact that it is a double helix. It's always a double helix. RNA, on the other hand, is going to be a single nucleotide chain. So it is a single-stranded molecule, and that single-stranded molecule is going to be able to take on a lot of different shapes inside the cell. Because RNA can take on a lot of different shapes, it will be able to do a lot of different functions inside the cell.